Tissue Engineering and Glycomics Research Assignment by Kelly Jo Guy, Cornerstone University. Tissue engineering utilizes living cells as building blocks. Examples include using living fibroblasts in skin replacement and repair, cartilage repaired with living chondrocytes, and other types of cells used in other ways. In these pictures, you'll see a tissue-engineered vascular graft, tissue-engineered heart valve, and an example of how cells are used in tissue engineering. Cells became available as engineering materials in 1998 when scientists at Guron Corp discovered how to extend telomeres, producing immortalized cell lines. Here's basic principles of the tissue engineering. You go in and have blood drawn or a biopsy. They send it to the lab. They expand the cells and they put it through a 3D polymeric scaffold, which grows a graft for skin, graft for a new organ, whatever the graft is. Research in bacterial glycomics could lead to the development of novel drugs, bioactive glycans, and glycoconjugate vaccines. Glycomics is very important in microbiology because glycans play many roles in bacterial physiology. While most definitions of tissue engineering cover a broad range of applications, in practice, the term is closely associated with applications that repair or replace portions of or a whole tissue. Bone, cartilage, blood vessels, bladder, skin, and muscles. In this first picture, you'll see an unraveling of the glycobiology. The second, in the middle, shows the tissue engineering of a urinary bladder. They are making a urinary bladder graft. And in this third picture, cells to be used in making a skin graft for a burn victim. In glycomics, there can be many challenges, like the complexity of sugars. Regarding their structures, they are not linear, instead they are highly branched. By the same token, glycans can be modified, which are modified sugars. This increases its complexity. Complex biosynthetic pathways for glycans. Another challenge is the usually glycans are found either bound to protein, which are glycoprotein, or conjugated with lipids, which is glycolipids. Unlike genomes, glycans are highly dynamic. I love this little cartoon that his blood sugar is high, but his salt, pepper, ketchup, mustard, and grated cheese levels are just fine. Here's a list of some of the different important functions of glycans. Glycoproteins found on the cell surface play a critical role in bacterial and viral recognition. They are involved in cellular signaling pathways and modulate cell function. They are important in innate immunity. They determine cancer development. They orchestrate the cellular fate, inhibit proliferation, regulate circulation and invasion. They affect the stability and folding of proteins. They affect the pathway and fate of glycoproteins. And there are many glycan-specific diseases, often hereditary diseases. These views here are views of how our family tree can determine our health. I want to thank you for watching my video. Here is my references, and I thank you very much. Bye for now.